Hi everybody, my name is Brad. I'm a PhD student here at the University of Southampton, and I'm going to talk to you about the undergraduate projects that I did here within the last 4 years. So in my third year I looked at autism. We did this using C. elegans, which are nematodes or worms. We knocked out receptors that we think are implicated within the disease in humans. We knocked them out of the worms, and we then observed how this changed their behaviour in terms of sensory integration. And then moved on to my fourth year project, which was my master's project. This time I used a different model organism called Drosophila, which are common fruit flies. You can see here I have a vial of Drosophila with food at the bottom. These are a really powerful system for many reasons, but mainly, or the main one for me anyway, is that they have a high genetic tractability, so we can manipulate their genome very easily and express things that we want. So for the instance of my project, I wanted to express human tau within the Drosophila and then see how this moves throughout the brain or propagates over time. There's a theory that tau can spread between neurons when it misfolds or before it misfolds within Alzheimer's disease, and this misfolded tau may be one of the main root causes for Alzheimer's. So when we're working with these flies, we anaesthetise them using CO2, so we have a CO2 gun here, then we put them on this pad which is slightly porous, and it keeps them anaesthetised. So if I look through the scope now, I pick two flies out. I picked a male and a female fly, which will show you what they look like here. So the top one is male and the bottom one is female. This is important, so we can take a female with a certain genotype. So for instance, this female can express a given protein in a specific tissue. Then we can take a male which has a specific protein that you want, so in this case it's tau. We had human tau with a fluorescent protein called M-cherry tagged onto it. When we mate them together, the progeny that come out will express your human tau with your cherry tag in the specific tissue that you wanted, because you had that driver in the female. So within this we get a specific protein expression within a finite area, and then we can watch this move from that area over time. So in order to do that, you take the fly heads off and you dissect them using basically just surgical gauge tweezers. We can then fix the tissue, and we can stain it with different antibodies. We then image it using a confocal microscope, which is essentially just a microscope with a laser on it that excites different wavelengths for you. And the finishing product we get here is something like this. So this is a Drosophila brain imaged on a confocal microscope with a stain for active synapses, so you get a good makeup of what the actual brain looks like. This is what it looks like at 0 days with the tau. So this red is our tau with the M-cherry fluorophore tagged to it. You can see here we expressed in both these areas. And at 0 days it hasn't moved far from that site of expression. At 14 days you can see there's already a great deal of movement from the tau. It's in these higher order brain regions that are connected up to the area that we expressed the tau from originally, and yeah, there's a great deal of spread. So this is just an example of one of the things that you could do if you did a master's degree or anything like that here at Southampton. As I said, I'm now moving on to a PhD, so I am using Drosophila still, but I'm instead looking at healthy aging and how neurons may change within a healthy system. This might tell us a bit more about mechanisms that go wrong in neurological diseases such as Alzheimer's and Parkinson's. Thank you.